My topic is biotechnology and society. What is biotechnology? My definition is very simple. So, biotechnology, it deals with application of biological knowledge and techniques pertaining to molecular, cellular, genetic process to develop significant improved products and services. Because see, here the topic is biotechnology and society. So society has certain demands and it has to be supplied. So the demands will be in the form of either a product or as a service. So how do you serve it? How do you supply it? So the supply will be the what vehicle will be biotechnology, and the methodologies, the techniques which the biotechnology will be using is molecular or cellular or genetic process. Okay. So in turn, because the demand and supply, it need not be only in economics, also in all the fields. This has to be matched. So there should be an equilibrium between demand and supply. Mismatch will lead to an increase in one phase towards the others, against the others. Okay. So biotechnological products have ensured it has ease of living, improved healthcare, agriculture output, created livelihood opportunities, innumerable. Because when I go through these slides, you will come to know we have done many things, or we have encountered many things which we are not aware. But there is a significant intervention by biotechnology. Okay, so by considering these things, there are basically, if we want to divide biotechnology into major segments, so it might be biopharma, very economically very more strong, more robust. Okay, then bioservices. Okay, and bioagri, with nothing but a biotechnological intervention in agriculture, bioindustrial, bioinformatics, and which together contribute to bioeconomy. Okay, so the digital economy, as we tell, so there is a bioeconomy. So it is very robust, and in nowadays it is catching pace and attention of various players. Okay. Yeah. So according to National Biotechnology Development Strategy, so the document says that India is ranked among the top 12 biotech destinations in the world and ranks third in the Asia. Okay, so we have our own unique position. So it is not that we have got it overnight. We have earned it over many decades of perseverance and efforts of many agencies, especially like DBT, DST. Okay, they have put very hard and efforts to bring this sector to what it is. Then the segment, this biotech industry is prospering because one is the economic prosperity of country is moving, our GDP is moving, and our per capita consumption is moving. So that one, because of that, we are having a growth in biotech industry as well as health consciousness. Nowadays, people, because of high earning power, okay, high purchasing power, we are more skewed towards health consciousness. Okay, we are ready to pay, but we want good health. So because of these things and our purchasing power and population base, okay, so the domestic demand is huge. Okay, so the unique position of India is, so if we are targeting the population in India itself, we will be serving to a larger population, and we can earn profit, as well as we can build a very robust sector in this arena. Then, so biotechnological industry per se, if we are speaking, in 2017 it was 44 billion market. In 2025 it is 150. You can just see from 2017 it took 6 years to reach 85, means double. But within 2 years it will be doubling to 150 around. Okay, such is a catapulting position which biotechnology has reached. Okay, so there are around more than 3,500 biotech startups in India, and within 2 years it may reach up to 10,000. Okay, by 2024-25. So having said these things, so history has to be reminded, because by we should if we know means biotechnology how it has grown, so we should credit to the contribution of microbiological sector. Why? Because to know biotechnology, it is very complex discipline. We want minutest particles or else in miniature versions. Okay, so microbiology turned out to be our candidates. Okay, so Chaim Weizmann in 1917, he was the first person to use microbiology for manufacturing cornstarch using Clostridium astobutylicum. It was for a different purpose to manufacture acetone for preparing atom bombs during the First World War. So the same person, Weizmann, became the president of Israel as well. So 1928, Alexander Fleming, he developed penicillin. So you people know, might be knowing that in our childhood was means an antibiotic which was means it was omnipotent, means it was everywhere, means any problem it was penicillin. Nowadays we are having many other antibiotics, but penicillin still dominates the medical industry, okay, because of its unique position. And this can be contributed to the basis of microbiology. Okay, then come Paul Berg's gene splicing technology, okay, in 1971. But landmark, because see, biotechnology is not a very cheap affair. Okay, there are, and it is a huge investment which is required, and something has to be pumped continuously. So for pushing the budget, the companies, they will be willing to have their rights, that is patents. Okay, so till 1980s it was not clear, but Supreme Court of United States, it ruled that genetically modified organisms can be patented. From then the GMOs, the biotechnology, it got a fillip. Okay, so in slides we will come to know various technologies whereby there are companies which have earned billions out of this biotechnology. Okay, so and major success of biotechnology lies in the legislation and enforcement of improved intellectual property rights, because biotechnological products, they are unique, they are patented, and the patents are worth crores. Okay, so you might have heard about recent means Covaxin, Indian vaccine, Bharat Biotech. Okay, so they're having patents of various vaccines, especially like Ebola vaccine, like that. So they are supplied to African countries, they earn crores. Okay, basically I am proud to say that Krishna Ella, the founder of Bharat Biotech, is an agriculture product, alumni of GKVK. So he turned from agriculture towards the biotechnological field. Now Bharat Biotech has carved out its own niche area. So IPR patents, they actually dominate the space. They are the fundamental stones, okay, base for the success of biotechnology. Biotechnology, if you see, there is a traditional biotechnology or conventional biotechnology, and there is a modern biotechnology. So out of traditional biotechnology, modern biotechnology, it stands for cell fusion, transfection, transformation, conjugation, GMOs, DNA and RNA things. Okay, so if you are going to apply biotechnology, okay, so in societal things in general, if we speak, there are all the sectors, sub-sectors, they are color coded. Okay, so this is it was published in 2004, and it is well versed thing. Nowadays we use this with synonymous thing, means red biotechnology, nothing but an health medical diagnostics. Yellow means food biotechnology. Blue means aquaculture, coastal. Green, everything to do with agriculture and environmental things. Brown with arid zone, desert. Dark means dark symbolizes something very negative. That is why dark, it is bioterrorism, biowarfare, biocrimes, anti-crop warfare. Then purple, patents, publications. White biotechnology, it is gene-based biotechnology, gene-based industries, it is enzyme industries, it is pharmacological, pharmacogenetics industries. Okay, gold, bioinformatics, nanobiotechnology. Gray, classical fermentation and bioprocess technology. Okay, these are the color coded various color coded areas of biotechnology. Okay, so biotechnology, specifically speaking, it is divided into various sub branches. Okay, one is as I told you, traditional biotechnology, traditional genetics. It is nothing but normal hybridization and selection. So I being an agriculture graduate, so hybridization of plants. It since millennia. So we are using this technology. So betterment of the better parent, all those things, what Darwin's theory told, what Gregor Mendel told, all those things, those principles are the rock solid principles based upon which the genetics and plant breeding is built upon, and the hybridization selection is based upon those things. Then mutation and selection is possible by traditional genetics. Then molecular biology, recombinant DNA technology, recombinant technologies, which helps in antibiotic production or else in new antibiotics, gene therapy also, all those things. Sequencing technology, forensic detection, gene banks, then DNA synthesis, then control gene expressions, antisense technology. So there was a question about antisense technology. I will explain because I'm having a slide about antisense technology. So cell culture. So means about apart from molecular biology, cell culture, cloning of organisms, or else monoclonal antibodies, stem cell therapies, all those things. There comes under cell culture. Then bioinformatics, application of information technology to biotechnology. Okay, this is a very potent field whereby crores and huge investment is going on this field. Why? Because it is having immense potential to grow. Genomics, proteomics, metabolomics, microarrays, computed drug design. Because now people want, because see, you might have noticed, a normal vaccine takes 5 to 6 years to develop. But the COVID vaccine, we got it within 6 months, 8 months. So this was possible because we had a prior information and prior experience and supported by bioinformatics. Okay. So proteomics and the knowledge of proteomics. So we could able to generate vaccines which could save millions of lives. Okay. So my first candidate is gene therapy. So you might be knowing gene therapy. So a gene therapy, a gene is a basic element in a DNA. A gene defines a character. Okay. It is a unicellular gene. It is okay. Multi means if it is a multi-gene characters like skin, hair color, or else an eye color, it is a different thing. So uni means if a single gene, single character. If some disease is occurring, it is because of some defective gene. Okay. So the gene has to be corrected. So how to correct? So it is gene therapy. So gene therapy, it can be either direct delivery or else it can be cell-based delivery. So direct delivery is in vivo, means you develop an gene and ready the dose and you directly inject. Okay. So for example, to liver, it is hemophilia. So hemophilia A and B. So they are the target cells are liver, muscles, fibroblasts. Okay. Cardiovascular diseases, it is endothelial cells, muscle cells. Okay. It can be in vivo or else it can be ex vivo. Ex vivo means you generate it outside, you take the cells out, so you insert the gene and you cultivate it, and again you are going to insert it. Okay. So gene corrected human pluripotent cells will be added. Okay. So this is gene therapy. Then stem cell therapy. Stem cells means, see, either it can be pluripotent or else it can multipotent cells. The stem cells, they are originate in the human body, either in the bone marrow or somatic cells, and stem cells can also be, they can be forced to become induced pluripotency can be induced to them. So stem cell therapy, it is having its own unique advantage. One thing is that through stem cell therapy, you can address various diseases like blood, like leukemia, then spinal cord injuries, skin burnt, so then liver, pancreas, diabetes. So all those things, it can be addressed. Okay. Various diseases are there. So the diseases can be addressed by using stem cells, which can be generated, which can be utilized to develop particular organs. Okay. Organelles. So it can be repaired. So another area is drug discovery. Okay. So you can see here the other picture. So here somatic stem cells are there. They are then they are induced to pluripotent cells. Then here disease specialized cells are there. From here, if you see, you can give it to two, means you can split it. Either it can be organoids or else it can be to drug discovery. Okay, drug screening. So because see, if you, when the drugs are developed, they cannot be directly tested on human beings. Okay, there is a phased wise trials. Okay, basic and clinical trial is basic. First, the laboratory confirmation, then clinical trials, then human trials. Okay, so we go for cell line cultures. Okay, for to know whether if I am having, one of my student is working on pomegranate seed oil. Okay, we are actually expecting to it to perform against pancreatic cancer. Okay, it has given a promising results. Okay, so without giving the dose to humans, only with using cell line culture, we could establish that it can work for pancreatic cancers. Okay, so first we have established now. Now after that, it has to go for hepatoprotective property of with cat, all those things, then followed by human trials. That is the last. Okay, so clinical trials, human trials. So here the drug discovery, because people, they want the things to roll out very quickly. Okay, so for to happen that one, we require analogs. Okay, so which has to mimic the things. So cell line cultures, here organoids can be there, which can be subjected, cell activity can be recorded based upon the drugs. New drugs can be founded and then it can be given to the humans. The alternate thing is that gene can be there, it can be corrected. Okay, it can be what healthy specialized cells, it can be rejected. It can be re-injected to the human beings. That is known as autologous therapy. Nowadays it is coming very fast. Okay, because means inserting good genes again back to the human body, so that you can correct the issues arising in the body. So the next concept is personalized therapy. Okay, personalized therapy, because nowadays if you see in society, various diseases are there which were present since many decades. Okay, but actually when we see, there are many of issues which is attached with that disease. For example, cancer. Okay, so earlier you see that any cancer, any colon cancer, pancreatic cancer, any cancer, if you see, what they used to do is that standard treatment. So any number of patients, if they are there, the therapy was to be there, it used to be uniform. Okay, when the therapy was to be uniform, then only few people which are over in green, you can see, they used to get good effect. Okay, the others, they used to not get any effect. Okay, the others who are actually at a negative side, they used to get an adverse effect. Okay, the therapy is generalized. Okay, so when it is generalized, it is common to all. But see, human genetics is very varied. Okay, it is diverse. So the genetic makeup of people are different. So the cultural background of people are different. Okay, the environment in which they are there, they are different. Okay, so all those things, it demands personalized treatment. Okay, so you can see through which personalized treatments means, basically they get biomarker. So biomarkers will be there, then blood, DNA, urine, tissue analysis, anything. So you establish certain things. Okay, then afterwards you can see it here. So cancer patients are there, then tumor, for example, then it goes for sequence. Okay, so after means picking your sample, it will be subjected to the sequencing. This is the role of bioinformatics. Again, marriage of bioinformatics with respect to pharmacogenetics. Okay, so here it is analyzed, then it will give. Okay, so what can be a drug? So you can see here green. So earlier therapy was only single drug. Here it is a combination of drug. Okay, so a simple drug, same drug if given, they will accept. It is effective. For these people, you have to have a combination of effect. Okay, the other, it is different combination. Then these things, when you match, then the result will be targeted. It will be personalized. Okay, so you might see that the chance, the success rate Earlier, in earlier treatments, it used to be 1 in 1,000. So, or else 1 in 100, like that. Now, due to personalized treatment, it will be minimum 90-95. Well, it has to be like that because huge investment is there. Okay. So, market size, if you see global precision medicine market, okay, in USA or else in Asia Pacific region. So, in 2019, it was 200. In 30, within a span of 10-11 years, it will be 730 billion of industry. Okay. Because see, it is nothing very fascinating things. In 80s, in 90s, it would have been like that. Post to 1990, post 2000 era, the science is moving in a very speedy manner. Changes, the changes are becoming very normal things. Okay, because see, if you might have listened about robotic surgery. Robotic surgery was very much new in few years ago. Nowadays, if you go in Bangalore only, 2-3 institutions, medical institutions, they are conducting robotic surgery. Okay. Then genetic gene therapy was not there. In Bangalore only gene therapy is there. Personalized therapy is there. HSG, HCG. Okay. So it is already there. Okay. This is a personalized therapy. Diabetes control. Okay. So what I am telling, see, these are fields whereby biotechnology has actually contributed, which is not visible, but it is what quantifiable through the data. Okay, diabetes very normal. We think that diabetes is not an disease at all. It is an invisible disease. Why? Because only 1 in 2 adults will come to know he is having diabetes. And but if you see the leading cause of death in the world, it is highest is it diabetes, 1.6 million. WHO, according to WHO, followed by HIV/AIDS, tuberculosis, malaria. Okay, so diabetes is the one thing which is causing too much of deaths. Okay, why? Because it is invisible. We are not aware that we are having diabetes. Why? Because we don't have screening mechanisms. Okay, so global prevalence in 1980 it was 47, and in 2014 it is 8.5%, 4.57%. Now it is 8.5%. India, okay, very alarming data. In 2015, it was 70 million. In 2040, and I saw recent data 2045, it will be 150 million of diabetes, double. Okay, the diabetic patients will be double. Okay, and see the spent amount. What was the amount spent? Rupees 90 billion was spent on diabetes management in 2015. Okay, so this is where society concern is. The society is concerned. Why? Because we are not able to manage diabetes and we are spending more. The amount spent towards any health care is drawing amount from somewhere else. Okay, so thereby we have to target and we have to create awareness about the management. Okay, if you see the human insulin, originally Genentech, a company, okay, you can surf it. Genentech is doing fantastic results. They are into basic genomics things. They are earning in billions. Why? Because their the technology is solid, fundamental things. Okay, this Eli Lilly company, what now it is present. So Eli Lilly in 1982 they got the first time the approval to develop the human insulin, that is known as Humulin. Still, these are the people who are dominating the market. These are the three companies who are dominating the market: Sanofi, Novo Nordisk, and Eli Lilly. Okay, if you see the journey of the insulin market or as the diabetes, the thing, it goes from 1922, isolation of insulin by Banting and Best, for which they got the Nobel Award. Okay, Banting got the Nobel Award. Okay, so after insulin was isolated, new generation insulin. Now we are here. Okay, next coming innovations once week, because you can see daily the insulin has to be injected, so it cannot be possible. So now means once in a week it will be coming. Okay, oral insulin will come. So vaccination, no more needles. Okay, so beta cell replacement will be there. So all those things, they are the future. Okay. So diabetes control is bitter truth, but it has to be controlled. Okay. So biotechnology is playing its own role. Why? Because insulin earlier it used to be extracted from the pigs. Okay. So we cannot have that much of a quantity because the population, you can see prevalence 4.7, it is increasing to 8.5, and the population people, they are 1 in 11 people are there having diabetes. For these many people, we cannot have so many animals to be slaughtered to get the insulin extracted. So nowadays, whatever the insulin is developed, it is BT enabled, biotechnological enabled. Okay, E. coli, Escherichia coli is used. The next concept is genetic screening. Okay, Madam was telling about prenatal screening. So we were very much unaware. Okay, because see, you are few teachers, you might have come across. So I am around 37, 38. So I was, because even though I prepared for civils and one of my option was anthropology. So for during my anthropological studies, I was knowing these things. Okay, genetic screening, genetic diagnosing, all those things. Honestly, when my wife got pregnant in the first trimester, they to ask me to go for genetic screening. Okay, so at that time I remembered, okay, because see, my sister just around 7 8 years ago, so she got pregnant. You might you can remember the things. At that time, no genetic screening, no genetic diagnosing was there. But nowadays, it is new normal. Okay. So for genetic screening, you have to go for genetic, means risk problems. How do you go for genetic screening? Maternal blood serum will be taken, or else prenatal ultrasounds will be there, or else non-invasive prenatal testing will be there, NIPT. Okay. NIPT, they are going to use the biomolecular, the markers will be used. Okay. Again, it's a blood test. Okay. It is non-invasive. So what it is going to give you? It is going to tell you whether there is any chromosomal aberration in the fetus. Okay. And if it is there, Down syndrome. If it is trisomy 21, or else Edwards syndrome, trisomy 18, Patau, trisomy 13. So this is screening, only screening. You are not certain. Okay. At least you will come to know that there is some issue, there is some possibility of an issue in future. Then you go for diagnosis. Okay. So diagnosis is either chorionic villus sampling and is amniocentesis. Okay. So amniocentesis chorionic villus sampling is this is known as CVS, which is commonly called. They actually confirm the problem of intensity of a problem which is there. Okay. So these things, what is the utility? Whether society has got any benefit out of this genetic screening. It is a basic biotechnological tool. What is the benefit? See, you might have known on a few years ago, a movie called as Pinky. It got nominated to Oscars from India. Okay. Pinky was about cleft palate. You can see this diagram. Okay, the picture of a boy who is having a cleft palate. Okay, and malformed encephalopathy. It is a Down syndrome, congenital heart disease. This is neural tube defect, NTD. Okay, so these things are not known, which we are not aware. Okay, earlier it was not accessible, available, or else affordable as well. But at least genetic screening is nowadays affordable. 500, 600 rupees, the test can be done. Genetic diagnosis, this is somewhat costly. Okay. So due to the competition in the field, now it is also made affordable. Okay. So if you spend 40, means all these 5 tests, it can happen in 35, 40,000 rupees. What is the utility? You can know many times, you can get a few slides from few WhatsApp sharings, Mila. So means the child has born with a defect. So you please contribute. So 16 lakhs, 20 lakhs, 1 crore. Okay. So what is the trauma of the children, means the parents to be there. So it is very unbearable. So this is an utility. Okay. Genetic screening. Forensic science. Forensic science, it is a pure application of biotechnology, nothing but an concrete evidence of a makeup of DNA. Okay, so you know DNA fragments. Nothing comes from nowhere. Something has to come from somewhere. This is a common concept in the biotechnological things. Okay, so it has to come from somewhere. So individual tested means if anything is there, forensic science, nothing but an it is nothing but a medical legal cases, MLCs. Okay, if you want to adjudge the disputed parentage, if you want to adjudge the identify an body or else an culprit, sexual assault, rape, anything. So forensic science is an answer. Okay, so individuals, they will be blood sample will be taken, DNA is extracted, then it will be sliced using restriction endonuclease enzyme, then smeared on GNA will be agarose gel, southern blotting technique, then radioactive probes will be used, and it will be x-rayed, then you will get an homologous chromosome. Everything is established. Now you will get a chromosome, you will match with your specimen thing. Okay, then if any matching is there, then you can certainly tell that it is because see blood group. Okay, blood group, if blood group is there, you cannot certainly tell he is the father. Okay, or else she is the mother. If the DNA fragment is there, definitely you can tell 100% because it based upon VNTR, variable number tandem and repeat. So at segment, the thing is that it is unique. Okay, so unique to the individual. I just wanted, because see, it is the case, it is about and in 2021 July last year, they solved a case. They solved this case about sexual assault, and they took 32 years to solve this case, and purely again it was solved based upon DNA fingerprinting. Okay, and what was the DNA which was used? 120 picogram. Only that is the very low quantity, and that to 32-year-old the DNA, the fragment was used. Okay, so that is one utility. Then if you see the NCRB data, see NCRB data, they tell me in 70,000 cases in 2020. Okay, they have been let off by the police. Why? Because lack of evidence, as well as the the crimes against women around 700, 1,200. Post Nirbhaya also. Post Nirbhaya also, they are let off because of insufficient evidence. Okay, so and many of the, because NCRB tells that around a lakh body they are unidentified in 2013 to 15. Everything it is because there is no proper DNA mapping is not there, or else the bank DNA bank is not there. Okay, so thing is that there these discipline has to flourish so that it can serve the humanity society at larger things. Evolution studies. So we know how we have evolved from a single cell to multicellular, and very from raw towards very refined things. Okay, to know these things we require basic and concept of biotechnology, biology, then the molecular biology, all those things, through which still you can see that many will be tracing our origin to our ancestors, because and these things will be used again. They are a very rich data. Okay, they can be used in bioinformatics to predict the models. Okay, so genetic models. Then comes monoclonal antibodies. See, monoclonal antibodies, nothing but an monoclonal. They are cloned. They are particular of a same variant. Okay, they are antibodies in general. Okay, so they have their own purpose. The purpose might be different. Okay, so it can be growth factor receptor blocking. So like anti-EGFR, inhibition of antigenesis, inhibition of growth factor, immunostimulation. Okay, so the induction of ADCC binding to FC receptor. Okay, or else induction of adopsis, or else antibody delivery, all those things. See, monoclonal body can be used to cure disease, or else to deliver the drugs. Okay, or to even to increase the immunity. It is more commonly, it used to be used for the thalassemia things. Okay. So nowadays, but it was again into news in 2021-22 due to COVID. You can see that emergency, the clearance was given for monoclonal antibodies. Okay. IgG, IgM. Okay. So immunoglobulins. It speaks because the monoclonal antibodies, COVID monoclonal antibodies, they go and bind to the receptors. Once they bind, the virus cannot come and bind to that same receptor. Okay, thereby you are preventing something from happening. So this was monoclonal antibodies. They are having huge market. You can see in 2012 it was 58 billion. In 2017 it was 98 billion. Okay, now it might be double that. Okay, I'm having a data of 2017. In 2022, minimum it will be around 200 billion due to again COVID. Okay, so you can see the application part. The monoclonal antibodies, it is synonymous with cancer treatment. So hence around 15, it is aligned with oncology or hematology. Okay, dermatology, skin cancer, or else thalassemia things. Okay, then biotechnology and vaccines. So you know that vaccines they are developed by using the E. coli or else the organisms, and it can be injected. So either it can be because we now are more aware about vaccines due to COVID things. But what is the biotechnological intervention? Okay, so the vaccines which are available nowadays, even the COVID vaccine is derived from microorganisms. Okay, rDNA technology, recombinant DNA technology. Okay, they are involving external microorganisms. Okay, so it is not because see, once whatever which is external is not having a ready acceptance in our body, we need an suitable mechanism so as to have an organic sync of the proteins. Vaccine is what? Vaccine is containing certain antibody. It is nothing but a protein. Okay, so the protein has to be given to the body. How you give the delivery, it depends. Okay, so basically there is a concept called as edible vaccines. Even Medicago, a company in Canada, they are in the third phase of developing edible COVID vaccine. Okay, so they are in the third phase. They are doing it. They are transferring into tobacco. They are transferring into aloe vera. So they are doing it. Okay, so edible vaccines, something means RNA, the genes are there. They are bombarded by using gene guns, vector system, chimera into the tissues. Okay, then it will give oral delivery to the human. Then the body will get the protein. Already in 2017, there was a paper on the hepatitis A vaccine, which was inserted into banana, and it was tested positive, means it gave a good result. Okay, so futuristic vaccines are edible vaccines. Okay, so because there is no end, there is no the scope, the limit for the creativity. Okay, next, biotechnology in agriculture. The biotechnology in agriculture has a very long journey to go because it is having immense potential. Okay, so we can use it for either disease-free planting material, fortification, reproduction of aquaculture, pest-resistant crops, biofuels, enzymes, fermentation, improving anything. Okay, n number of things are there. So if you see the basically animal breeding, pulse breeding. Okay, then conservation of germplasms, plant breeding, single cell proteins, tissue culture. These are the main arena where we are working. Okay, so there is a concept called as single cell proteins, SCP. Okay, SCP, single cell proteins. Nowadays you can see that there is a protein deficiency, malnourishment is there, kwashiorkor, marasmus. So those things, they are protein malnutrition. So we require proteins in a very crude form, very concentrated form. Okay, so for which we require certain entities who can give it in a very easy manner, speedier manner. Okay, one such thing is spirulina. Then other microorganisms are there. You can remember now the CF they came up with spirulina chicky. Okay, because spirulina is a very condensed form of protein. Okay, So the spirulina, any number of ACP, single cell protein, this is nothing but a microorganisms, or it comes under the field of industrial microbiology, okay, whereby the microorganisms, they are genetically engineered and they have been made more efficient. They are grown under fermented tanks, then purified, then extracted, then given in a form, either a powder form or as a paste, whatever the end user, okay. Commercial biotechnologicals, biotechnology, commercial means markers will be there. You can use markers to identify certain traits, okay. So vaccines, tissue culture, I'm doing, yeah. Tissue culture plants. Tissue culture basically means extracting a tissue and culturing a plant out of tissue, okay. Tissue culture, okay. It can be meristem tip culture, or else it can be an somatic hybridization, anything, callus culture. So applied, deployed, n number of opportunities are there, okay. Because the tissue culture has established as an itself as a very refined field whereby immense potential is there. So you can see various stages, tissue is taken, grown, then multiplication stage, callus formation is there, then root, then shoot, then acclimatization is there. Afterwards, transport means transferred into pods in a nursery. So for what what are the purpose for which we can use tissue culture? Clonal propagation, disease-free planting materials, wide hybridization, early flowering, synthetic seeds, fast multiplication, somatic hybridization. Then Madam was telling about tomato. It can be done. Applies, pure lines. If you want to develop hybrids, you want a pure lines. Again, tissue culture. Okay. There is another field, secondary metabolites. Okay. Nowadays, metabolites, they are of immense pharmacological value. Okay. Azadirachtin. It is present in neem. Okay. But it can also be developed by using tissue culture. Capsaicin in chili. Vincristine in Vinca rosea, Cathanthus roseus. It is an anti-cancerous property, vincristine. 1 kg or else very means even 10 grams will cost you lakhs of rupees. Okay. So by growing rose means Vinca rosea, Sadabar, common name. Okay. If you grow, you can extract only 0.0001% of vincristine in a kg of rose petals. So that is highly inefficient. Then what is the efficiency? You can go for plant tissue culture. Tissue culturing and enabling the mass so that you can extract the vincristine out of it. Sterols, stilbens. Okay. So there is no end. Okay. The secondary metabolites can be produced in a very speedy manner, in a refined manner, in a clean and hygienic things. Okay. I wanted to share a story about tissue culture. Okay. You know that commonly tissue culture tubers means the potato tubers. Common propagation mechanism is tubers. Okay. What happens is that this story is real and it is happening in Karnataka, and we are having this collaboration with the International Potato Center. This is my university logo. We are having a collaboration with CIP Peru. So basically the farmers face a problem with respect to the quality planting material, that is seed tubers. Every year, if you people are aware about the growing of Assam region, if you belong to Assam region, you will come to know, every year they have to get seed potatoes from Punjab for sowing. So it is having a high cost of seed material, around 60 to 75,000 rupees per hectare, and 40 to 50% of the total cost of production it will be same. So if that is the thing, if I am not getting a good planting material, if every time I have to get the potato from the Punjab, then how it is possible? Then there is a technology called as ARC, okay, apical root cuttings. So by tissue culture, you are going to have root cuttings. Again, that root cuttings will generate into 7 to 8 tubers. That's tubers can be, again, you can directly go for cultivation, or else it can be used as a seed tuber. Again, multiplied and used in further things. This will bring the production cost to 25% of what earlier it was. From 75,000, it will come out to 25,000, less than that. Each tuber will only cost, planting material will be less than 1 rupee, highly profitable. So this is now government of Karnataka is also promoting this in Assam areas. So tissue culture, if you see banana, potatoes, sugarcane, apple, pineapple, strawberry, there are many this one who are based upon tissue culture only, especially banana. So if you see, our bananas are exported, and I credit it to the tissue culture things. So the tissue culture market now, it is around 500 crore rupees. Even DBT has started quality certification for tissue culture. Okay, it is a unique thing, but it is having a 100% compliance from the company people. So GMOs. So what is a GMO? Genetically modified organism. Okay. Here comes first GMO crop in the agriculture is flower sour. It is tomato. So it is about delayed ripening. So there is a question I told that I will answer. This is antisense technology. Okay. Antisense technology means gene silencing, antisense technology. You are going to silence the gene so that it will not express. Okay. RIN and NOR gene, they are present, they are to be silenced. If you silence, so in flowers or tomato, what happens is that if you see, it will be firm when you buy it. As the time goes, it will become less firm. Why? Because in cell wall, there will be polygalactonase enzyme. Okay, polygalactonase enzyme will break down the pectin, which is present in the cell wall, and it will make it more flexible, soft. Okay. Why? Because the enzyme will act. It will act. It is known that it will act. But if we delay the action of that enzyme, then we can delay the ripening of tomatoes. Means it can be kept for more days. The shelf life extension, minimum keeping quality, can be increased. So such antisense technology was used, and the variety was flower sour. Then came 95, canola, composition, all those things. In potatoes, glyphosate resistance in US. Then comes potato ring spot virus, dominant in papaya ring spot virus, dominant in papaya. It actually eroded the huge plantations. Right now, the papaya has grown in US. They're all GMO. Means they are inserted this potato ring spot through using a gene gun, a gold enabled gene gun. So thereby it is now GMO. Okay, they got a very good results. Golden rice, you can see. Again, I will talk, because golden rice, earlier madam was telling that golden rice 1 and golden rice 2. This is the 2000, it is Inga Porticus IRRI, International Rice Research Institute, Manila. They developed golden rice in 2000. IRA, it developed in 2008-10, like that. That was golden rice 1 of India. Then now there is a golden rice 2. The golden rice 1, we made a mistake. What? We got a gene, the yellow gene, from daffodils. Okay, daffodils is non-edible flower. Okay, we understood the problem. Okay, we realized the problem. Then we went for golden rice 2. That is again golden rice only, but the source is not daffodils. The source is Zea maize. Okay, now maize is used as a source for golden rice, and the amount of the keratin, beta carotene in golden rice 1 and amount of golden rice 2 is 4 times more. So GMO crops, again coming back to GMO. So these are the many biotech crops which are grown throughout the world. Now most of around 70 countries, they accept the GMO crops. So in India, you can see we only grow cotton. Cotton is very dominant. You can see USA, around 37% of their area is GMO. Okay, around 35%. So canola, around 95% of canola is GMO. So if you see brinjal, it is only in Bangladesh, not in India. Why? Because we nearly reached of developing BT brinjal. Again, it was the same, having the same bollworm of cotton. Same Lepidopteran was infecting the brinjal. But thing is that due to GEAC, Genetic Engineering Approval Committee, they told no, and we went for voluntary moratorium. But Bangladesh has released the brinjal couple of years ago. Okay, but the thing is that we are not sure that whether these brinjals are coming to India or not. There is high certain that it will come. Okay, because it is a porous border, everything is uncertain. Okay, so 70 countries they are growing GMO crops. You can see the map. So soybean is dominating the market 50%, followed by maize, cotton. Okay. Number of countries, if you speak, it is the cotton. Number of countries they are using cotton, maize, and then soybeans. But in USA it is soybean. Okay. So what is the utility of GMO? Increased crop productivity. Why? Because GMO are nothing but an hybrids. Hybrids give you good results. Okay. Biodiversity. Why? Because they will give you more crop per unit area. Hence, no land additional land is not required. Then better environment. Why? Because herbicide and the insect resistance, insecticide use will be less. CO2 emission. Why? Because it will save the production of the herbicides, insecticides, all those things. Okay. Then poverty, hunger, all those things. It is there. Okay. Advantage. I want to tell you the story of the BT cotton contribution to the society. Okay. So if you see, this is an cotton growing country, cotton producing country. Okay. This is India, green. Okay. This is black dotted is USA, and this is China. Okay. If you compare 1960, from 1960 to 2000, we are in the abysmal low. We are not a major grower of cotton at all. Okay. So it used to be China, followed by USA. USA is at the median. We used to be at this point. From this point, our journey started in a very catapulted manner. Okay. We have now reached, we have surpassed China. We are the largest grower of cotton in the world. Okay. So one is this story. Second story, the world market, world cotton market, if you see, in 2002, it is an inner circle. 12% is only Indian market. Now it is 25%. We capture 25% of the world market. Earlier China used to have 28, now it is 25. US 19 to 14. Other world decreased. The Brazil was also increased. Okay, but we are having a sizable chunk. From 12, we have gone for 25%. These two narratives are supported by this one. This is adoption of cotton, BT cotton in India. Okay, BT cotton, so how they are adopted. So if you see, BT cotton means earlier cotton area per hectare, okay, cotton production was there. But BT cotton was here. Just you take this graph and with you match this one, you will get the results. So BT cotton has given us fillip, okay? It has made India from net importer to net exporter of cotton. Okay. Now cotton, and again that data, if you see here, we used to, prior 2000, we used to only have cotton for our internal consumption. You can see here. Okay. Post 2005, we started exporting. Now we are one of the major exporters of cotton. So because BT is a biotechnological tool. Tool is simple. So what they did is Bacillus thuringiensis, Cry1 ACG from BT, they took and they inserted into cotton. So what it gave protection against bollworm, which was a dominant pest in the cotton. So for which they used to spray huge pesticides. Okay. So that was intended use. So if you see this graph, so the pesticide reduction from 46, it has reduced to 20. Okay. And especially the cotton insecticides itself, among total pesticides, from 33 it has come to 11. If we speak about the savings, from 160 million we are going to save, means right now only expenditure on cotton bollworm is 25 million. Where 160 million and where 25 million? 135 million is the savings. This is the economic saving. This is the export is the economic earning. This is environmental earning, means we are saving the things. And if you save the spray, the livelihood of the farmer will be improved. Okay. So there the livelihood, it is the proven documents that means here it is there. Kranti, he is the director of Central Cotton Research Institute, which is there in Nagpur. So he has given a very beautiful document about how it has changed the livelihood of the farmers. Yes, it also led to the suicide of the farmers. Why? Because in growing BT cotton in non-conventional areas will not yield a good results. Yes, but thing is that it has given a very phenomenal results. Gujarat is a major cotton growing area. it has benefited by BTA technology like anything. Okay, so we have benefited a lot from biotechnological tool in cotton. Okay, BT brinjal concerns are there. So I will tell at the last slide. I'm having certain things to share. BT brinjal, what is the issue? Biofortification. See, BT technology, all those things, we are inserting external gene into the things. So thereby what we are doing is we are making the things which are not normal. We are going to add additional. But we want natural. Okay? Why? Fortification is something which is adding additional nutrients into the organisms. So why? Because to address hidden hunger. Hidden hunger will lead to stunting, blindness, poor brain development, weakened immunity, anemia, all those things. So GDP labs will also be there. Why? Because the hidden hunger will lead to inefficiency among the population, will lead to the lack of its his or her contribution in the economical affairs. Okay, in India itself, the loss of GDP is 12 billion. Okay, in other countries there are millions. In India it is 12 billion. So address to that one, we are having biofortified. Biofortified means certain genes will be explored traditionally and they will be went with a conventional breeding. Okay, so basically they are targeting micronutrients as iron, zinc and vitamin A. So that is beans, rice, the iron in beans and pearl millet, rice and wheat, cassava and maize. So this is a Harvest Plus, a CIMMYT, a global agency which is working to address this biofortification. You can see here, you can see India has developed two varieties of the sweet potato. Boo Sona, keratin enriched sweet potato. Then Boo Krishna, that is anthocyanin enriched sweet potato. Pusa beta kesari. There is a cauliflower which is enriched with beta carotene. You can see here the maize. I told you that maize, Pusa Vivek, it is tryptophan enabled maize. Along with that one, beta carotene has been enabled. Okay. So here, if you see biofortification, so feeding the chickens with the biofortified maize has enabled to change. You can see the leg colors and you can see the change in the egg colors, egg yolk. Okay. So why? Because it is all about biofortification. Conventional breeding, conventional inserting the genes, desired characters. You can see in last year again 2021, 17 biofortified varieties were released in India. So oleic acid, which is very much required MUFA, it leads it is accepted. Means it is desired. So it is enabled in groundnut GMR 4 and GMR 5. So whereas low erucic acid in mustard. Erucic acid leads to the cardiovascular issues. So it has been reduced. So here the yam, so whereby anthocyanin has been enriched. Okay. So another applications of industrial biotechnology, white biotechnology, nothing but a fungal biotechnology whereby the fungals will be used to develop various elements like polypeptides, terpenoids, insecticides. See, fungal things, fungus can be utilized for anything. It only depends upon your the knowledge how to use it, because they are delivery mechanisms. You can see here enzymes. Again, fungal Aspergillus niger is used commonly in the food industry for production of enzymes like protease, carbohydrolase, lipase, esterase. So these things are not visible. If you see, you might have tasted appy juice. Okay, appy juice is clear. Why? Because they use protease enzyme. Was a pectinase enzyme, they use it for clarification. That pectinase enzyme is an industrial manufactured pectinase enzyme and it is manufactured by Aspergillus niger. Again, role of biotechnology, which it is invisible, which we don't know. Okay, it is an apple juice. Because if you see in your home, if you make an apple juice, it will be cloudy. But appy, it is carbonated, fizzy, and it is clear. Okay. If you see any juice, it will be clear, means they have used enzymes. Using enzymes is not bad. Okay, it depends upon what is the purpose for which we are using. Fermentation, we are using the wines. For example, the waste, horticulture waste is very much because see, if I take mango, okay, mango pulp will be only 50%. 50%, not 50%, 40% will be pulp. Remaining 60% is a waste. What I will do by taking that waste? Okay, apple pomace. After extracting juice, what I will do with the left out the pulp, that is known as pomace. Okay, so for those things, you can see that nowadays you can get fruit vinegars. These fruit vinegars are manufactured by using the strains which are biotechnologically engineered. Okay, even bananas, when they are too cheap, it can be converted into the ethanol, then acetic acid. Acetic acid nothing but vinegar. 5% acetic acid is a vinegar. Okay, so then comes wine. Wine is nowadays the per capita consumption of wine is increasing like anything in India. So we are becoming more what familiar with more wines, whether red wines or white wines, desert wines, okay, or also dry wines. So the thing is that malolactic fermentation is very much important. In wine, the enzymes and the organisms, they play a main role. Okay. One thing is that I will tell a common analogy. Wine. We use yeast for the Saccharomyces cerevisiae which we use, or else the wine yeast which we use for manufacturing of wine. It is having a tendency. So it can only, the normal analogy tells that if you use the ethyl alcoholic fermentation, 50% fermentation, it will give 50%, if 100 grams of sugar, it will give 50% of alcohol. So the efficiency is only 50%, one. Second thing is that wine varieties are having sugar content of 24 to 26. So the wine strains which are normally available, if they go alcohol beyond 10%, they cannot survive. There is no fermentation. So what is the answer? They came up with the strains, genetically engineered strains, which can resist up to 13, 16. You can see good wines like Grower Zampa wine, La Reserve, or else Vijay Amritraj collection, or else anything which are more than 1000 rupees, 900 rupees. If you see, alcohol content will be minimum 13 to 16. Minimum 13 to 16, it is not at all possible by using your dairy, means the normal baker's yeast or else normal yeast which is available. So because it cannot survive. Why? Because beyond 10 it will not survive. So along with that one, I want a good aroma and minimize the biogenic amines. So SO2 content should be less. When I filter wine, it should be having a pectolytic activity should be there. Wine should be stable. Okay. All these things is possible by using these strains, okay, the organisms. Then comes the concept probiotic, which madam has told. Probiotic, the thing is that probiotics, nothing but a live microbes, probios. Means whatever the microorganisms are there, which give beneficial things to us is known as probiotic, okay. So various organisms like the lactobacillus species are there. Then lactobacillus, bifidobacterium, all those things are there. If you see the history, one thing I want to stress here, the first stable culture of probiotic, Lactobacillus casei shirota, okay, 1930. He developed this strain. If you remember the now present Lactobacillus casei strain shirota, it is present in a major dominant probiotic product in the world as well as in India. That is nothing but Yakult. Okay, you can see here Lactobacillus casei shirota. That is a strain which was developed by Shirota in 1930. It was preserved. It is an patented strain. And whether they're having B Active is there, Amul Pro Life is there, Nesvita is there. What are they doing? Whether, see, probiotic is not normal curd. Even normal curd, dosa, idli, they're all probiotic foods only. But these probiotic things, they are added with the cultures. These cultures, they are not normal simple cultures, they are manufactured cultures. For example, there is a company called as DSM. Okay, they come up with a Delvo. Okay, because I did for one of my studies, we did probiotic strain of kokum juice, for which we imported this, we got this specific culture. Okay, why? Because to call it as a probiotic, you should require 10 to the power of 12 CFU counts. Okay, so billions of the organisms should be there. So probiotic, There is a lactobacillus bifidobacter is the one of the organism which is Indian, because probiotic is gut specific. For Indian, we should have a gut specific for us. So there is one means this is a one field which has catch the industry. Cheese making. See, cheese making is synonymous with respect to the enzyme called as the chymosin. Okay, chymosin or else renin. Earlier it used to be extracted from cow, but the cow calf muscles, it means it cannot be extracted. It is inefficient. So what we did is we started using the recombinant DNA technology. Then we put the chymosin strain, again multiplied, extracted, purified. Then we get the cheese. What is the market? Whether there is a market, there is a huge market. Right present, it is a high market here. Here is a medium, it is a low. We can just match with respect to the consumption pattern. The milk, we were not nearby. Before white revolution we were nothing, but now we are the highest producer of milk. Okay, per capita consumption highest in the world. So milk products, they are gaining because our diet pattern is moving towards westernization. Cheese is becoming new normal. Now pizza, pasta, all those things, it is nothing but a cheese. Okay, so for these industries, next big thing it is going to happen in this area, whereby the cheese market will be penetrated. The European, they love the cheese. They are means traditional. Okay, there is one bovine somatotropin arm. Again, rDNA technology. Basically, why I have this slide is to show you how not only the quality but quantity can be increased. Okay, so by inserting bovine somatotropin hormone, the cow the milk yield can be increased even to the tune of 20%, more than 20%. Whether it is safe, European Union is not accepting, in certain countries in Europe not accept, but WHO tells it is safe. FDA tells it is safe. Okay, so even the DuPont, they are having the permission to use this one because Eli Lilly was developed this one. They sold this to DuPont. DuPont is having one brand which is selling this bovine somatotropin hormone. Okay, there is a huge market for this one. Cloning, there is a concept called as cloning. You might have heard earlier, it was there. Cloning is what? It is nothing but a creation of very same to itself. Okay, so cloning is having very various issues attached with it. So why I want to what I want to highlight is that, see, cloning, here you can see here we got means various NDRI, National Dairy Research Institute, which is there in the what Haryana. Okay, so there they have developed this female calf Swarupa. Okay, adult buffalo. So you can see around 11 buffalo calves have been developed, out of which 3 have been died and rest are there alive. And this is a recent one. What Ganatantra and Karnika. These are the 2 indirect cloned cows are developed. Why cloning is necessary? Why? Because we are having milk production. I told that milk production is there. You can also remember there is a debate which is going on, A2 and A1 milk. Okay, so we require A2, which is now fad of the day. Means it is now the people wants A2 better health. So why? Because A2 is a protein, beta casein. Okay, A1 is a means another protein. A2 is a different protein. A2 milk is a milk which is produced out of indigenous cows. In India, it is developed out of either Gir, Sahavali, Dioni, Taparkar, all those things. Okay, so these cows, they have their own problems. The thing is that they are having A2 milk, good quality milk, but their production quantity is very less. So how to improve the breeds? Then we have to go for cloning, then hybridization. Cloning, hybridization, so that it has to match with quality and quantity. Why? Because right now, if we go for indigenous cows, okay, and if you tell that we follow indigenous cows, we don't want the HF or else and Jersey, we don't want. No, we cannot do like that, because the milk production, it cannot be profitable by using indigenous cows at present. It has to be made profitable by increasing the milk yield, milk quality. Okay. So cloning is the application. Again, biotechnological intervention, if applied in because government of India is having the conservation and promotion of indigenous cows in its program. Okay. Environmental biotechnology, there is a concept called as bioremediation, which you might have heard about. So bioremediation is remediating, giving remedy to some biological issues. Okay. So how to address? Because see, whatever the things are there, see, nothing is not, means everything is connected. In this world, everything is connected. If you do somewhere, you are supposed to bear the fruits out of your ill effects, means or else your actions. Okay. If you contaminate the landfills, if you contaminate the ocean, or else aquifers, all those things, because you're going to fish it, you're going to consume it, then you're going to consume the milk, ingestion, all those things. Anyway, the heavy metals, the, because I will tell you a concept. So you might have heard about green revolution. Green revolution, it gave agriculture phenomenal increase in production, but it had its own problem. Extensive use of fertilizer in Punjab led to nitrate toxicity in water. Okay, this nitrate toxicity has led to cancer cases in Punjab. There is a train which runs from Batinda to Jaipur. It is known as Cancer Express. Okay, so this cancer nitrate, then you can correlate to bioremediation. So these things, whatever there is in toxic levels, it has to be remediated. Either you can remediate by microbial, FICO, or else zoo, phyto, anything. Okay. But the thing is that we are having limitations. Database for contaminated sites, application, risk remediations, all those things. So what is the scope? If you see, this is a scope. What metabolomics, proteomics, genomics, everything, it is oriented towards biotechnology. Okay, it is a societal problem. Societal problem has to be addressed by someone, and that is science, and science should have a vehicle, that is biotechnology. Okay, if you see synthetic biology, bioremediation, all these things, they come under synthetic biology. So all the sensors will be there, gene editing will be there, microbial consortium will be there, synthetic bioremediation sites, all those things. But these are nothing but a cocktail of microorganisms. So these microorganisms will be given in situ or ex situ. Okay, and you will be having desired results. So one such thing is bioremediation of oil spills. You can remember that in Pacific Ocean, if many of the times the ships will be, they will be unloaded, means they will be having a failure, then they are going to have an oil spill there. So you can see many birds which are damaged by it. They cannot fly for a lifetime because of that oil spill. So what has to be done? There's a concept called as oil zapper. It is an Indian technology. TERI and the ONGC combined, they have formed OTPL. See, petroleum company, TERI energy company, they came together, they formed biotech company, OTBL, ONGC, TERI, Biotech Private Limited. Okay, so that is the company which manufactures your oil zapper. Oil zapper is a technology whereby it is a consortium of microorganisms. It goes, it binds to oil, it converts oil into CO2 and H2. That's the simplest form. Okay, so rest of the things, again, it can be utilized itself for its as a substrate. You can see here scientific data. Okay, so this is a Pirana in Gujarat, oil spill before and after. Then Kalol, Gujarat, before and after. So bioremediation, if you see bioremediation, GC fingerprinting, by using GCMS, so the alkane, the profile, you can see here, before bioremediation, the alkane profile gave the streaks like here. After, no peaks. No peaks means no alkanes present. Okay, you can see quantity of TPH present, and after 4 months, nearly nil. So bioremediation is an effective technology. So it is to give answer to the pollution problems. Okay, oil pollution. Here the plastic. See, plastic is nothing to, means it is very serious issue. So there's also called as concept called as microplastics, which are very serious than the macroplastics. So landfills, it takes around 400, 500 years to decompose the plastics. So at present, India is having only 11 kg per capita consumption of plastic at present. By 2022, it is an old paper article. Now, by this time, it will be 20 kg and 20 million tons by 2020. So if it is a case, then it will lead to various health hazards in us. Okay. So what has to be done? So biodegradation by mechanism. So again, microbial cultures will be there. They will the fragmentation process will take place. Oligomer to dimer, monomer assimilation. It will lead to again these byproducts, means what monomers are produced. They will be used by microorganisms as a substrate. They are going to give back methane and water, which will be escaped to the atmosphere. Okay, biofuels. Okay, this is national national policy on biofuels in 2018. See, biofuel is a very critical area which we are concentrating nowadays. Why? Because 70% of our oil consumption comes from outside. So we have to have a self-reliant, Atmanirbhar Bharat. So to become self-reliant, we should develop certain mechanisms to develop our acumen. So now we want to move from, see, there is generations of biofuels. Generation 1, 2, 3. Okay. Generation 1 biofuels are from food grains. That is the soya, that is the maize, that is first. Second, the biofuels from the non-food. Okay. So those are the things like waste edible oils, non-edible oils, used cooking oils. Okay. Short gestation crops. Second generation. Third generation, advanced biofuels. Okay. Third generation will be algae based biogenerations. Why? Algae can give you 10 times the productivity of these second generation biofuels. 10 times the productivity, thereby efficiency and effectiveness will be more. Okay. The means concerning to this, we established our own the biofuel park. First biofueling park was established by Dr. Ashwardhan Ji, Minister of India. Then Center of Excellence in Bioenergy has been developed so as to develop organisms which can synthesize certain enzymes which can increase efficiency in biofuel fermentation process. Okay, there is also called as animal diagnostics due to the intervention of the bioinformatics. So development of various technologies like Brucella free India, what we want, major disease in among the cows, or else a keto check, conductivity meter, LFA check, all these things. Bioinformatics merged with the veterinary medicines. Okay, there is very dark, I told you, dark biotech means dark area. That is nothing but a bioterrorism. Okay, we cannot ignore. This is a fact. Okay, because when COVID actually occurred on the global arena, first we thought that whether it is not and whether it is a bioterrorism an act by any neighboring country. It was on suspicion. Why? Because as Madam told in the previous lecture talk, it is a lab. It was escaped from a lab, so it is having a huge potential. Okay. Now bioterrorism, but it is not proven. It is only in the analogies, or it's only theory part. Okay, nothing is proven. We cannot blame anyone, but it is a possibility. Okay, so bioterrorism you can classify into ABC. So bioterrorism at a society, see, because there are many warfares which have been fought. Okay, in the First World War, they used the bombs. Okay, normal TNT bombs. Okay, in the Second World War, they used the atomic bomb. Okay, which was more potent, and with a single bomb, you know that what is the effect. Now the Third War, it will not be with the use of bomb. It will be use of the bio organisms which have been engineered so as to create a problem among the society at large. Okay, so the bioterrorism is a real possibility. It can happen, but the thing is that there are various checks. Cartagena protocol is there. Then there is various checks and balances, okay, to control those things, okay. So A protocol, A, B, C. A is most extreme, most devastating. It can take huge toll on death, like Ebola, okay. Then the bacillar anthrax, okay. Then the variola. These comes under A. B comes under alpha virus, brucella, so staphylococcus aureus, food toxication. Then comes the C, Nipah, Anto, flavivirus, Mycobacterium tuberculosis, all these things. C. So these three means the organisms. Yeah, the see in the global arena, the geopolitics. Everyone wants to be powerful. Everyone wants its his decision to be heard, and it can reach to any extent, and they can employ any methodologies. You can heard that because Arab Spring happened, because there was rumor that he had dirty bombs. Dirty bombs means the chemical warfare, which actually was used by USA in Vietnam Wars. That is naphthalene, all those things were used. Okay. Now, I come for pros and cons of genetic engineering or biotechnological. Pros, you know that I have debated: fighting disease, increased life expectancy, so all those things. Then coming to cons, the religious and ethical concerns. So cons means basically religious and ethical concerns, because see, I told that there is a bovine somatotropin hormone has been injected into milk, and milk production has increased 20%. So religious concern, whether it is a religious concern? Yes, it is against kosher requirements of Israel. So Jews, they don't use that milk. It is against the kosher. Okay, religious concern is there. Okay, ethical concern, yes, cloning, creating new thing is, and a copy, a Xerox copy of anything, it's ethical, against ethical. Okay, genetic issues. So genetic abnormalities can arise due to an improper use of technology, or also an exploitation of suitable vehicles. Then health issues, because health issues are allergies. Because drug resistance, because see, drug resistance or resistance, it is quite common. But if you are using the genetically modified organisms, so biotechnology, I'm not telling biotechnology general, is these things more or less it is attached with GMOs. Okay, allergies, because the external organisms are there. Okay, so I told you that with respect to vaccines also, vaccine allergy also will be there, side effects will be there. We also know in COVID we are having Covaxin, Covishield, Johnson Johnson, anything, Sputnik, everything is having a side effect. Okay, what is the side effect? What is the derived benefit? We have to see. Okay, so the allergies, health issues will be there. Resistant insect pest, yes, because BT cotton. BT cotton means Bacillus thuringiensis Cry1 AC gene has been added to cotton. If some pest consumes that one and it becomes resistant, then that is known as super pest. Okay, if weed is become resistant, super weed, that is quite natural. Okay, but we have to think, so that for that purpose only we tell that when you spray insecticides, spray in a limited quantity. Okay, that is quite possible. Antibiotic resistance, because antibiotic resistance is quite common. Reduction in genetic variety. Okay, genetic variety, variation. You know that there is a species diversity, genetic diversity, all those things. So genetic diversity is very much required in the global arena, in the as a environment per se speaking. Why? Because if it is not diverse, the things will go homozygous. It will become more simple. When something uneven happens, there is no diversity which is there, and everything will be wiped out. Okay, so certain refinements is not necessary, at least with respect to genetical makeup of any organism. So I now tell you why BT cotton, BT brinjal was not accepted. Because BT brinjal, it was the genes, BT genes, the candidate crops which were used, means candidates what germplasms, lines. One such germplasm was Matigulla. Matigulla, if you belong to Udupi, you might be knowing, indigenous to Udupi. It is geographically identified crop. Okay, so that is indigenous to that area. So there will be many such varieties, species which are indigenous. Okay, when you go for refining, okay, refining homozygosity, those genes are eroded. Okay, so the genetic diversity will be eroded. Okay, because if you insert Matigulla, means BT gene to Matigulla, then what happens? Matigulla will not be BT Matigulla, it will be BT Matigulla. It will lose its the genetical makeup. On effect on wildlife, same thing, soil pollutions, displacement of natural species, it can be there. Influence of certain industries interest group, it is possible. Why? Because they have a huge pocket, huge MNC. If you come across, most probably in there is a TV series nowadays coming as Human Trials. Okay, so web series you can watch. So there you can see the might of a company to determine the narration of any human trial of a drug. Okay, industry interest groups, because see these biotechnology should be an vehicle. It should be an vehicle to create simple technologies in a very affordable manner. Okay, but certain, for example, BT, I told you, there is a concept called as termination gene technology. Termination gene technology means if you purchase certain BT genes now, if you sow in your field, again next season if you want to sow, it will not give you good results, same results. Why? Because they are having terminator gene inserted, means the effect is terminated in that generation. So next generation it will not be there. Okay, so terminator gene technology is there. Many industries they employ. Why? Because they tell that we have invested billions of money in the R&D of this BT technology, this biotechnology things, all those things. Okay, so pros and cons are there. What is the final saying? The thing is that, see, I am having this photo which is very much visible. So the ball is having a double helix model of a chromosome, Watson and Crick. So everything it revolves around this the beautiful double helix model of a chromosome. So the gloves symbolizes the lab. Okay, laboratory, or else we can have technological intervention to n number of things. But thing is that, as we tell biotechnology, it should be right technology. Means it should be available, affordable, and accessible to all. Okay, it should create equality among all, and it should breed opportunities to many. Okay, it should not be causing any hardship to even a small community, because some communities they conserve the seeds, okay, for which we are having PPV and FRA legislation. Okay, so it should what have an equitable opportunities to all, equal considerations of all the opinions, and the world is fragile, because as Madam told, in a matter of 1 week everything goes up and down. Okay, so which we saw in especially in Karnataka in second wave. So the gloves, why? Handle with care. It is there, you can manipulate it, but manipulate for the good, not for the bad, for the betterment of the mankind. And mankind, we have to see the opportunities. Opportunities are innumerous. Okay, so having said so, thank you.